because we're seeing these dynamics across the brain, we're seeing intracortical connectivities or correlations, we're seeing these patterns that are just, glaring out there. And in fact, the physicists in us look at this and say, well, if you measure 200 neurons, you're not going to see the same thing as if you measure the average of 10,000 neurons. So it's certainly true. If you zoom in on them, you're going to find a whole bunch of cells that are doing a whole kind of milieu of different things. But when you average them all together, they're showing you an average over this spatiotemporal unit of an increase or a decrease in their ensemble behavior. And I'm just saying that from a shot noise perspective, it's actually much harder to calculate I always use the analogy of voting, right? If you asked 20 people on the street in New York City who they were going to vote for and then tried to predict the outcome of the general election, you'd be in big trouble, right? You need to have this average over this large population to get a sense of, in some cases, even whether activity is just going up and down. And the fact that we see this coherence, the fact that we see these little functional units and we see the way that they're correlated to other areas, way across the brain in what they're doing, I can't back off from thinking that's important. It's definitely super interesting to go in and then say, okay, of those maybe 10,000, 100,000, I don't even know how many neurons are contributing to that ensemble signal, what are they all doing individually and how does their behavior change from moment to moment? But the fact that they are exhibiting these ensemble properties, I find super interesting because it's something that I actually know people who are going in and looking at one two-dimensional plane with their 2-photon microscope and sampling 100 cells are struggling to predict whether the animal's whisking or not. Whereas it's just there for us, right? It's just glaring in those more average signals.
But I guess, Mark, to your point, it seems that it is possible to collect very large populations with close to single neuron data. From my perspective, and I think a lot of the people listening, it's hard to keep track of what's going on the optics side of experimental neuroscience because, recently there was a paper that came out on bioRxiv from Vaziri Group. It was looking at light bead microscopy and imaging of, it claims 200,000 neurons at 5 hertz. And you're like, okay, light bead microscopy, that's the way to go. But then we have these other great examples of wide field. Still, a lot of people are thinking, well, if you're in the brain, you've got to be doing 2-photon, it has to be multi-photon. And it's hard to keep track with so many different modalities where the state of the field is right now and what if you're interested just in population imaging, what should you build? And so that's what I'm trying to get a sense from this group, where is the field now? Is there, I know it's a lot to unpack, but can we make some generalizations about if in this situation, these tools are good, in these situations, these tools are good?
Before even trying to address the question, I think you raise a really good point, which is that there are a lot of options. So one of the things that I think that is, coming to the fore is that it's not just about cellular resolution. We also have to talk about the fidelity of the activity traces and to what extent, our scanning method, for example, may compromise photons that may compromise the resolution of various types of spikes. But the larger conundrum I think that you're raising is very much true. There's a whole bunch of new techniques, and it's not easy necessarily for people who've not been trained in optics to decide what's the right match between the question or set of questions that I'm interested in and the optical techniques that I might consider, maybe none of which are commercially available at the moment, right? In electrophysiology, there's obviously a range of tools. Intracellular recording approaches, whole cell, extracellular. For some reason, people seem a little bit more comfortable, deciding which electrophysiological techniques they might use. Not always, there obviously are some subtleties there, but particularly in the optics domain, there's a limited number of groups that build their own setups and many more groups that I think maybe in the position that you described, right, of trying to figure out, well, which direction do I go?
So Matt, I'm going to push back on you a little bit because this is a pub thing, right? So I'm going to pretend like we're actually at a pub and you're not recording this. So I, to some extent, object to this line of questioning in the sense that I think counting the number of neurons is, I'm going to speak out of my depth. I just think it's the wrong way to think about imaging, and maybe historically comes from the world of electrophysiology, because that's what you could do, right? You could measure spikes, and I can make a probe that can measure from 10 neurons. I can make a bigger probe that can measure from 12 neurons. But the thing is, we're actually not talking about what we're measuring. So those extracellular electrodes measure action potentials, right? And so that's what we count, right? We're like, oh, I can measure action potentials from 100 different neurons. But what we're talking about here is imaging. We're not even measuring action potentials for the most part, right? We're measuring calcium, which we believe to be correlated to the average number of action potentials over a period of time. And at what frame rate, right? So these aren't even one-to-one comparisons. And I think, we can speculate as to why that is, but I think it's very hard to, try to compare the raw numbers of neurons that you're getting from an electrical measurement to the raw numbers of neurons that you're getting from an imaging experiment and try to make, comparisons between the two.
I think even someone who's relatively nuanced and sophisticated about this, they're still saying like, okay, I want to understand population dynamics and motor cortex. I'm willing to make trade-offs. Like I would potentially be willing to just look at, single neurons with 100 millisecond bins, sparsely sample, get, couple tens of them. I'd be happy if I had an optical approach that averaged over potentially signals from hundreds of nearby neurons if I understood, exactly what I was getting and I could build a model around that. But I think that many people don't even know what they could get from optics. They just fundamentally don't even know what's available.
I'll pat in here. Okay. So firstly, there are lots of courses. I'm part of AQLM. I'm part of the Cold Spring Harbor Imaging Structure and Function in the Nervous System course. There's lots of places where people can go and learn some of these fundamental concepts. I think the first neuroscientists came from electrical engineers because they had to figure out how to do electrophysiology. So I don't think it's okay to just sort of say, well, it's not fair because biologists can't understand the optics. I mean, there's some fundamental principles here that are perfectly understandable. I think on the flip side as well, there's a lot of math and a lot of modeling and a lot of things that you really truly have to understand because you can get yourself into a right mess, even if you get all of this imaging data. In terms of the imaging technologies, I haven't been blowing my horn here, but we have a technology that is, really good at doing exactly what you said. My approach has always been that actually we have to have a technology that is simple enough for people to be able to actually use. And I think one thing that tends to happen is you get technologies that get more and more and more and more and more complex to become more and more and more trying to do more and more and more things. But then in practice to actually use them, to implement them, they're very, very expensive. They're very, very difficult to actually get to work. And I think all of that is driven by this mindset that you're coming to this with, which is saying, tell me what to build. And that's the other thing. And I'd like to bring Mark in on this because you said, tell me what to build. And Mark and I have had this conversation before. And this is really hard for us as technology developers because yes, Bell Labs built the first 2-photon microscope. 2-photon was licensed, so nobody could actually use a viable system, so everybody started to build their own 2-photons. And I would tell you that building a point scanning 2-photon is easy. I've had undergrads build it. We build them in a week on these courses. It's not that hard, right? But this expectation that people have in neuroscience that they can just build it, I think is also one of the really big problems. So I want to raise all of those points and see what my friends have to say.
And even electrophysiology is moving in the direction where you can't just build it. You can't just build your own neuropixel anymore. Right.
If I could just jump in and maybe support both what Jacob and Elizabeth are saying. So to Jacob's point, and I think we've sort of danced around this a little bit, but let's just bring it to the fore. We're in the age now where we have a great deal more knowledge about cell types. That this is undoubtedly one of the major achievements of the Brain Initiative has been the elucidation, at least from a expression perspective, the different cell types. And so if we want to be able to understand what the new plethora of cell types are doing, we have to have recording technologies that are up to that job. And one of the beautiful things about imaging is that we can genetically target, sometimes in multiple colors or with other tags, the cells that we're interested in, try to dissect actually what different cells are doing. And it can also be done by projection pattern, right? So I think one of the stunning things, if you just visit the Allen Brain Atlas, is all the different projection patterns that you can just browse through.
I think that the thing that's holding neuroscience back right now is this divide between, well, I'm an experimentalist and you develop instruments and you do the analysis and we don't need to understand each other, right? And I've always joked that having divorced parents was great experience for me and doing interdisciplinary science because I got very used to having to explain to one group, it's okay, they didn't mean that. It's, try to explain it there and then go back and explain it on the other side. So I've always sat in the middle. And I love the fact that I'm always learning, right? I'm always trying to understand what is the biological question that this person's trying to tell me that they want to understand. And then I get into it, and then I actually want to study that myself. And then, so I think we should look to a future where you don't have someone that identifies, I'm a neurotechnologist or I'm a neuroscientist, but that we just train the next generation of people who are trying to understand the brain in all the kinds of things that you need to know and you need to learn to be able to leverage these technologies. Whether it's to improve them, or the other thing I would say is we need to fix the culture. I don't know, again, if Mark and Jacob fully agree with me, but I still think that there's a little bit of a disconnect in appreciating the complexity and the value of the technology piece compared to the neuroscience piece, and sort of leveling that playing field so that you really can get these collaborations. So it doesn't have to be one lab saying, I need to just build your microscope and scurry off and do some experiments, but that, we should have equal numbers of people with these different diverse skills working together in groups and having a real focus on the results that we want to get.
Well, I certainly agree with what you were saying about the collaborative aspect of things. Elizabeth, there's one thing you said that made me a little nervous, and maybe I was misinterpreting it, but that people should maybe think less about specializing and calling themselves, say, theorists or experimentalists or technologists. And the reason that makes me a little nervous is that I look at what has worked in traditional physics disciplines, like particle physics, where it was actually very helpful to have different groups of people who were specialized and focused on different things, and they were collaborative. And so I really like that aspect of it. And they were sort of on equal social footing, if you will. But having domains of expertise where there really were people who were allowed to specialize and to gain the proficiency that specialization permits was really critical for the advancement of particle physics. And I tend to think that I think we do need a culture change in the sense that we need to honor people who are specializing and provide them routes for career progression and allow them to be on, say, on equal terms within a collaboration. But I do think that as we go forward and technologies become more advanced, that specialization is going to remain important.
I agree. And if I look around at my lab, I always explain this to people. I say, I want you to come out knowing how everything around here works, right? If you tell me I'm a biologist, I'm not changing that hard drive. Like, no way. You've got to be able to, do all of these things. But everybody has a natural talent, or they come from a background of some level of specialization, or some people just like doing some things more than others. And I fully embrace that. That's no problem. It's just the silo. It's just that's not my business. I don't have to learn that, or I don't want to learn that. I think people need to understand that, to be able to do this stuff, you have to. I mean, all three of you know how upsetting it is when somebody's done some analysis on their data and you have to tell them that it's just not right. It's just noise. They've misunderstood, right? And, we just need to get past that because, otherwise, I mean, if somebody tried to do computational imaging by putting a piece of tape on the front of their camera and collecting a bunch of data and being like, yay, Jacob told me this is going to work. Like, that's not going to work. Right. So we've got to just, I think, embrace that training.
I take a slightly different perspective because I think my field more closely aligns with neurotechnology than neuroscience. But if we were to place the words, I think it's very similar. What I heard from you, Elizabeth, a little bit is that it's important, I think, for the technologists to really deeply understand the problems that they're working on, and vice versa to some extent, right? The people who are designing the next set of experiments have to have some appreciation for what's possible from the technologies that are available, or what might be possible. So I think that the interdisciplinary training obviously is important. And I guess maybe what has worked reasonably well for us is for the technologists to work side by side with the people who are trying, basically your user, right? So however you define your user, if that's a neurobiologist, that's a clinician, it's incredibly important that you work with your user. I think the danger that we run into is when engineering gets divorced from either the problem or the user base, and then we start making stuff. We're like, I can make something that can, meet some performance metrics, but if that's not important for your user, I'm not sure that that's a good use of our time. And maybe the trade-offs that you made in building your next widget really make it so that it's not actually very practical. So if you have the opportunity, young neurotechnologists out there, find places where you can work with the neurobiologists, the clinicians, and so that you're tailoring your work to solving a problem.
And teach them and learn from them. Get that exchange of knowledge. I would agree. I mean, the things that have been effective in our lab has been when you've had really equal buy-in from both groups and you've really had people side by side working together, seeking to understand